Okay, this is lab exercise number 10, hearing and equilibrium. Looking at the objectives, you're going to describe the structures of the ear at gross and microscopic level. Discuss the structures of the ear as they relate to the functions of hearing and equilibrium. Look at the Rhine test, the Weber test. Compare and contrast conduction and sensory deafness. Look at how low and high pitches are localized, discuss vertigo, and look at vestibular nystagmus. Okay, the ear is much more than something that collects sound waves. The ear is a receptor organ for two senses, hearing and equilibrium. Hearing is the function of the outer ear, middle ear, and inner ear, while equilibrium is a function of the inner ear. You're going to look at two different hearing tests, the Rhine test and the Weber's test. You're going to look at how to localize sound. You're going to look at equilibrium and vestibular nystagmus. Hearing, kind of like in Chapter 9, is the perception of the sound. It's an interpretation of the sound vibrations. The pathway for hearing is at the pinea. The sound wave travels through the external auditory meatus to the tympanic membrane, to the eardrum, Tympanic membrane oscillates about the width of a hydrogen ion, and the vibrations are conducted to the amplified to the auditory ossicles, the malleus, incus, and stapes. The stapes pushes against the membrane of the oval window, and its oscillations set a fluid called perilymph in motion. Know the difference between perilymph and endolymph. The movements of perilymph cause the movement of the vestibular membrane, whose movements are transferred to the endolymph and to the basilar membrane. The downward deflection of the basilar membrane to the basilar membrane and to the tectorial membrane. The physical stimulus of the vibration is finally transduced to a nerve impulse. Hair cells synapse with the auditory nerve, which synapses with the inferior colliculus. From there, the impulses are transmitted to the thalamus, then to the auditory cortex of the temporal lobe, and finally, sound is perceived. Damage to the tympanic membranes or auditory ossicles result in conduction deafness. That's conduction, so you actually have a physical damage to parts of the ear. Damage to the inner ear or to the auditory nerve results in sensory deafness. That's a lot harder to deal with. Conduction deafness, we can push the sound waves through the bones with a hearing aid. If you've got sensory deafness, you're pretty much deaf. Vibrations can be conducted by bones or the skull, especially through the mastoid process. We'll see that when we do our tests, our hearing tests. You can amplify these vibrations. That's where a hearing aid comes in. That is for conduction deafness. Two tests that we're going to look at today, they're going to be the Rhine and the Weber. They differentiate between conduction and sensory deafness. The Rhine test, you're going to take a tuning fork, tap it, put it by the mastoid process, wait till it goes away, and then if you move it back in front of the ear, you should hear it again. That would be that would say that you not have. If it didn't come back, you would have a conduction deafness because the vibrations are being picked up by the mastoid process and kind of pushed into the ear, kind of a roundabout way. If you move it away from the mastoid process out to the external auditory meatus or the outside of the ear, it doesn't come back, then you've got some sort of conduction deafness. In the Weber's test, you put the tuning fork over the mid-sagittal line, and what would happen is you would hear it louder in the deaf ear. You would hear it louder in the deaf ear because in the non-deaf ear you're going to get a lot of noise from the room, and so you would not hear it as loud in that ear. The normal ear would interfere with the hearing of the tuning fork. A person with sensory deafness would, however, hear it louder in the normal ear. So in the Weber's test, you've got it over the mid-sagittal line, the tuning fork, because again, it's being conducted through the bones of the skull. With a conduction deafness, the sound is still getting there, and you're going to hear it louder in that ear because of the interference from the sound in the other ear, the room sound in that. With a sensory deafness, the only way you're going to really hear it is in the good ear. So if you've got a bad ear with sensory, you're simply not going to hear it, or not hear various wavelengths. Conduction deafness is kind of for all the wavelengths. Sensory, you can have some interference with specific wavelengths, i.e. you can't hear high-pitched sound or various pitches. We're going to look at the localization of sound. Localization, here's another part of the inner ear. Localization of the sound takes place with two methods. One, with the sound waves. A lower pitched sound has a much longer wave, a higher pitched sound a much shorter wave. Two ways you're going to look at the sound is the lower pitch sound will hit one side of the head and then wrap around and hit the other side. Again, it's milliseconds difference, but your body can tell the difference. You'll hear it first with a lower pitch sound. You'll hear it first in one ear and then a millisecond later in the other ear. That will give you an indication of where the sound's from. You would turn and try to locate it at that point. With a higher pitched sound, it tends not to wrap around, but the intensity of the sound, you hear it louder in the ear nearest to the sound, and then less loud on the ear on the other side. And that's why you kind of turn your head until you can get a fix on it. Perception of equilibrium involves different receptors. You're looking at the vestibular apparatus, the eyes. You have proprioceptors in the joints and tendons that help perceive position. Neural pathway integrates and interprets this information in the cerebellum and the vestibular nuclei of the brainstem. Movements of the eye and body are controlled in response to this positional information. The vestibular apparatus consists of the semicircular canals and the utricle and saccule in the inner ear. The structural difference you can see here on figure 10.3. The structural difference you see there. Both structures, the receptors are hair cells, which are bent as endolymph moves through the membranous labyrinth as the head is moved. The utricle and the saccule, perceive linear acceleration or static equilibrium, allowing us to remain upright, hold our heads right, hold the upright position of the head. The semicircular canal maintains dynamic equilibrium, and here's your semicircular canals. You notice they go in all three dimensions. The semicircular canals, you're looking at dynamic equilibrium. The hair cells are embedded in the gelatinous membrane of the cupula. The semicircular canals are oriented in three different planes: sagittal, frontal, and horizontal. Rotation sets the endolymph, the fluid within the canals, in motion. Initially, the hair cells bend. That's when you feel the acceleration or the motion. Once that initial inertia is overcome, the hairs, and everything is spinning at the same rate, the hair cells bend up, and then you don't feel it anymore. And then when you stop, again, the hair cells, the body is stopped, the semicircular canals are stopped, but the fluid inside them is continuing to move. And then they're bent in the other direction, and that's where you get being dizzy, or you get what they call, again, the vestibular nystagmus. It's a result of the suddenly stopped rotation. We didn't do it in the lab. We may still try to do it. And if you start spinning and the endolymph catches up with the rotation and then you stop, this is conflicting. This causes the eyes to move in the opposite rotation and then jump back to center. And this is part of the vestibular nystagmus. Now then, this also looks when you're looking at motion sickness, because in the brain you're getting two different types of input. You're getting the input from the eyes that say, "Hey, I'm not moving." If I'm reading a book in a car, you're getting input from the eyes that say, "I'm not moving," because there's the book, it's right in front of me. Everything else, the hearing, the vibrations, is telling you that you're moving. Sometimes the body, some people just can't handle the two different sensations, and they're much more prone to motion sickness. Okay, so let's look down here 1 second. You're going to do the Rhine test, the Weber test, your localization of sound using the high and the low pitch fork. Again, the low pitch wraps around, you hear it one ear then the other. High pitch, it's in its intensity. Somebody who can't hear with one ear is going to have a lot more trouble, because they're going to be unable to localize, because there either isn't the difference in intensity or the time difference in the sound arriving to the ear. You're going to need to look at vertigo, which is a dizziness. You're going to look at Meniere's disease, which is an accumulation of fluid. You look at tinnitus, which is a ringing in the ear. Okay. Here is, you're looking at the head. This is figure 10.15. You're looking at with the head up, the otolithic membrane. As the head is bent, the stereocilia bands change. They give different sensations, and you know that you've bent your head. Endolymph in the semicircular canals, your cupula, hair cells. As the head turns, you can see that the hair cells turn, and that gives you the sensation of rotation. Vestibular apparatus, joint tension. It goes into the cerebellum, back to the vestibular nuclei, the brainstem. You've got the oculomotor center and the spinal cord, which controls eye movement and head movement. Excuse me. Okay, I think we're going to stop right here. Excuse me 1 second.